Hello, my name is Christina Austin C. I'm a clinical molecular geneticist at Mass General Brigham and the analysis team lead at the Center for Mendelian Genomics at the Broad Institute. And I am here today to lead us into the genomic analysis hands-on workshop. So I will first present some background on some basics of analysis, a pretty high-level overview, as well as give an introduction to using seqr. And then we'll be sending everyone to breakout rooms to try your hand at analysis yourself, of course, with some guidance. So we're really looking forward to this session. So today we'll be focusing on the analysis of exome and genome sequencing data. So collectively, these two techniques are referred to as genomic sequencing. And they both produce quite a bit of data. So in exome sequencing, you're sequencing all of the protein coding genes in the genome, which is about 20,000 genes. And this will include both known disease genes and genes that have no known association with human disease. These genes would also be sequenced if you were going to do genome sequencing, but in a genome sequence, you're also including the vast amount of non-coding sequence within our genome. So it's only about 1 to 3% of the genome that counts for coding regions. So in a genome sequencing, you're going to sequence virtually all of the DNA, about 3 billion base pairs. So what you get from these techniques, once you've gotten your sequence off the sequencer, aligned them to reference genomes, is about 50 million reads in an exome, which will contain about 100,000 positions where your patient sequence will differ from the reference genome it's aligned to. In a genome, you get 1 billion reads and millions of variants. So overall, this is a lot of data to deal with and can seem a bit overwhelming at first. And of course, the challenge to anyone doing this analysis is, of course, how do you find the few disease-causing variants and genes in this vast sea of data? And it's filtration that makes this work manageable. And this is the step we'll be focusing on primarily in this session. It's the process by which an analyst will apply a series of criteria to narrow the scope of variants requiring manual review. These might be things like population frequency cutoffs, inheritance patterns, specific variant impacts, that ultimately allow the analyst to narrow down to, on average, the 1 or 2 variants that might be of interest to the patient under analysis. So in order to be able to filter variants by things like population frequency cutoffs, you first need to go through a step called annotation, which is bioinformatics step, where you actually append each variant with a large amount of information. So the example shown here is a single variant. We would append, the gene impacts, the transcript impact, what kind of variant it is. So in this case, a canonical splice site variant, the zygosity of the variant, some quality metrics to assess how high quality the call is, population frequency data, things like whether it's been reported in association with disease before. So this is a really critical step in the exome analysis process. So once you have a well-annotated data set, how might you go about actually filtering data from, for example, an exome sequencing case? So the truth is there isn't actually great standardization in the field about how filtering is done, and filtering should be tailored to the specific goals of what you're trying to find in your case. Are you looking for novel gene discovery, or is this a clinical case, for example, where you're trying to find a diagnosis, something that's clinically actionable for a patient? So let's run through a very simplistic example to prove the point of how this might work. So here we have an 18-year-old male that had recurrent oral ulcers, and an exome sequencing was ordered. Notably, this patient also had a strong family history of these recurrent oral ulcers, so there was some suspicion that this was a genetic defect. The first step you might take to try to narrow down the scope of variants from the 67,000 that were identified is, as I've mentioned already, use a population frequency cutoff. So here, the problem we're trying to address is that we all carry a lot of variants that are far too common in the general population to be disease causing, so the example shown here, while 60% of the general population has an A at the position that's highlighted in red, 40% of the population has what would be called as a variant at that position, an A to T change. But given that 40% of the general population has that change, it's very unlikely to be disease causing. So we use large population data sets like NOMAD typically to filter based on frequency, and I know I'm not going to spend a lot of time on NOMAD as Anne has already introduced it, but this is a quick screenshot of the type of data you might find in NOMAD, and it can be a little tricky, to be honest, to kind of decide what an appropriate population frequency cutoff is, because it depends partly on the frequency of the disease you're looking at, the penetrance of the disease, its inheritance pattern, but a commonly used cutoff in exome and genome sequencing is a 1% frequency cutoff. So if we were to apply that 1% cutoff in our example case here, you can see that you've gone from 67,000 variants down to just 7,000. So that's a massive reduction already in the number of variants you might have to look at, but still 7,000 is really not feasible for us to manually review. So what might you do next? Another commonly used filtration method is to filter based on quality metrics. This is getting at the problem that not all variants called by bioinformatic pipelines are real variants. I'm showing an example at the right here of what a real variant looks like. We have a C to D change with an approximately 50-50 allele balance present on both forward and reverse strands, which are distinguished by the red and blue coloring here. In contrast, you see some of the other calls in this region are present in only 1 or 2 reads, and these reads often have multiple changes. So these are some of the hallmarks of a false positive call that we don't want to be spending time on in our analysis. So I'm highlighting on this slide 2 different quality metrics that are available in seqr, so paying particular attention to those, though there are many others that can be used. The first is allele balance, which is representation of what fraction of the sequence variants contain the variant. So for a heterozygous change, this would be in the range of 50% or 0.5. However, there is a range of what's observed in the sequencers. So we tend to set a more permissive cutoff of 0.2, below which you'd have a low-level mosaic change or a false positive call. The second metric that we're talking about here is a genotype quality. So this is a measure of the likelihood that the genotype call, homozygous reference, heterozygous, or homozygous alternate, is correct. The values of this metric range from 0 to 99, with 99 being a high quality variant. We usually set a cutoff of 40, and this is just based on our own particular pipeline. We have taken a look and demonstrated that most variants under a call quality of 40 are false positive, though there are exceptions to that. So returning again to our case example here, if we started again with our 7,000 rare variants and we applied a genotype quality cutoff of 40 and allele balance cutoff of 0.2, we get down to 4,000 variants. So again, we've significantly narrowed our scope, but this is still too much to look at. So what's next? So this is where I like to think that analysis becomes a bit of a choose your own adventure, right? So there's a number of different approaches you can take at this point. And surprisingly little standardization in the field as to how this should be done. But you know, the specific approach you take really should be tailored to your goals, whether you're doing a clinical analysis, or more of a novel gene discovery type analysis. But overall, the best approach, I'd say, for the most part, is one that combines several different strategies. So I've listed out here kind of 3 major buckets of strategies that we think about when we do analysis. So these include variant-centric approaches, phenotype-centric approaches, and inheritance-centric. And I'll start with the phenotype-centric because I think it's often the one that most people think of. It's a very logical approach where you're saying, I'm going to develop a list of genes that are most likely to be relevant to my patient phenotype. and then just screen all of the variants within those genes. While this concept seems fairly straightforward and logical, it's actually harder than you might think to define what the list of genes are that are most relevant to your patient's phenotype. So there's not really a single source, unfortunately, in which you can just type in a phenotype or a disease and get a comprehensive list of all genes that are associated with that disease. So how do we go about generating these lists when we want to do a phenotype-centric filtration? Ultimately, we end up making gene lists that are combined from a number of different sources. So we might include variant databases like ClinVar and HGMD, the Human Genome Mutation Database, which is unfortunately not free to use, but is a fairly good database of variants that have been published in association with disease before. We also will take gene disease reference materials like Gene Reviews and Medline Plus, both of which are better resources for more well-established disease genes. OMIM is a database of gene disease associations that tends to capture more newly discovered genes, though is certainly not perfectly up to date. Panel App is a service, kind of a crowdsourcing approach to developing gene lists that has been developed by Genomics England. And actually, it's a really nice new feature that we have in seqr that we have imported all Panel App gene lists into seqr automatically. So anything that's available from Panel App can be used within seqr already. And GenCC, which I believe has been mentioned earlier in this workshop, but is kind of the ClinVar of genes, if you will. It's a database of gene disease curations from participating laboratories and can be a good source of interesting genes you might want to look at. And then over on the right, the Genetic Testing Registry is a database of clinical tests based in the US, and this is an important resource. So if there is a clinical test available for the phenotype you're looking at, you'd obviously want to make sure you're screening all the variants in those genes. And then PubMed, sadly, a lot of novel, or rather newer gene disease relationships are really only captured in the literature. So they're not yet structured in these databases like OMIM. So it can often be useful to do a brief literature search for the phenotype of interest to be able to capture the newest gene disease associations for that disease. So returning to some of the other approaches you might take, the variant-centric approach shown on the left here is where you're saying that you want to look at all variants that are most likely to be classified as disease-causing. So generally this is an approach we call a genotype-centric approach, where you want to prioritize variants based on the suspicion of the variant and then evaluate whether they're relevant to your patient's phenotype after they're returned. And so there are a couple of different buckets of variants that we would normally return in a filtration like this. The first major bucket being variants that are predicted to be loss of function variants. Now, certainly not all loss of function variants are pathogenic. There's a number of genes where loss of function is not a known disease mechanism, right? But these types of variants can sometimes be classified as pathogenic with little other information depending on the gene they fall in. So this is a bucket of variants that we generally want to review in cases. The second major category in this variant-centric approach that I would look at would be variants that have been previously associated with disease. So this would be, for example, your ClinVar pathogenic, likely pathogenic variants. And also sometimes we use other variant databases. So the challenge in that step really is that the most of this gene or variant disease association is in the literature, which is highly unstructured. So we are dependent on variant databases to pull this kind of information out. Another one that is commonly used but is a proprietary database is HGMD, or the Human Gene Mutation Database. And on the right here, our last category is the inheritance-centric approach, which is similar to a variant-centric approach in which you're prioritizing variants based on their inheritance pattern and then reviewing if they're of relevance to your patient. So this is particularly powerful if you have multiple family members sequenced, in particular a trio, so when you have both parents and an affected proband. In this case, you might consider looking at all de novo variations, so all variants that have occurred in the proband and are absent from both parents. If a recessive inheritance pattern seems likely for your patient, you could also look at all the homozygous calls below a certain frequency cutoff, or all compound heterozygous calls, so all the genes that have two rare variants in them would be something you might want to consider as potentially interesting findings. So if we were going to go back to our case example and just select one of these approaches, I might apply a filter to look at only variants, these high-quality rare variants that are classified as pathogenic or likely pathogenic in ClinVar. And in this particular case, if we were to do that, we would end up with 6 variants to look at. So now all of a sudden we've gotten to a point where there's a manageable number of variants to manually review. Generally, the first part of this manual review step is a very high-level review of all of the variants returned by the filter. It's sometimes called triage. And in this review, an analyst is really considering multiple different pieces of information at once. You're asking, does the variant have sufficient evidence to be considered disease-causing? Does the variant fall within a gene that has an established association with disease or falls in an interesting pathway for the disease you're looking at? And then, of course, does that disease, that pathway, have any relevance to your patient's phenotype? So there are multiple different pieces of information being synthesized here. And this is a fairly time-consuming process. So the ultimate goal of your filtering strategy needs to be kind of striking a balance between capturing all the variants that you might be potentially interesting to your patient, but also limiting the number of variants that need this very time-consuming manual review. So if we were returning to our case example here, we've narrowed it down to 6 variants. It may be that the answer is found within that, but this is in fact a very limited search. We're only looking variants in this last step that have been detected by another laboratory, which is often not the case for most rare disease variants. This variant, even after being detected, had to be classified as pathogenic, or likely pathogenic, and also had to be submitted to ClinVar by that laboratory. So that's a very, very small subset of variants, and this would not represent a comprehensive search. And as I mentioned previously, really comprehensive search approaches, generally the best approach is to take multiple different approaches. So an example here of a kind of combined approach that we might apply would be to include previously published variants, so ClinVar path and likely path below a frequency cutoff. We're usually a little more permissive with that cutoff to account for things like founder variants or high frequency known pathogenic variants. We might also filter in the predicted loss of function variants below a 1% frequency cutoff. Sometimes you limit these to medically relevant genes, particularly for clinical cases. And those would be genes that have had any association with human disease in the past. Another block of variants we might bring in would be, again, like a phenotype-centric approach, looking at all variants in a gene of interest. So this would be the way you'd bring in maybe a previously unpublished missense variant that you might want to review. And then in the last box here, the coding and splice region variants with interesting inheritance patterns. So this is where you might look at all the de novo variation or all the homozygous and compound heterozygous variation. So at this point, hopefully you're feeling confident and that you have some idea of how to approach analysis and you're excited to try this out on your own. There are many different analysis platforms available for exome and genome sequencing, but today we are going to be using seqr, which is an analysis platform that was developed in our group at the Broad Institute. And this is an open source platform that is designed to enable collaboration and open data sharing. It integrates many of the data sources I have referenced earlier in this talk, and hopefully you'll agree that it has a very user-friendly interface. And if after participating in this session you think that seqr might be useful for your own work, I do want to remind you again that seqr is now available on Anvil. So any Anvil user can bring your own joint called VCF or create one using Terra-supported workflows, and then load that data to your own private seqr project for analysis. So I'm going to switch over now to a live view of seqr to show you some of its features and walk you through basic analysis steps. So the first thing you'll have to do when you reach seqr.broadinstitute.org is to log in. You should have pre-registered with seqr prior to beginning this session. If you were not able to pre-register, that's okay. You won't be able to do live analysis in the breakout rooms to follow, though of course you can still join to watch. But I will post the registration instructions in the chat, and we can always get you access to try your hand at analysis later this evening. If you have registered and you have some trouble logging in, please email at the contact us link here, seqr.broadinstitute.org, and we'll try and see if we can quickly resolve that for you. But hopefully you won't have any issues. You'll go to the website and click sign in. It may ask you for the credentials, the email address you registered with. I just have mine saved already in the browser. This first page you'll see upon logging into seqr contains a list of all of the projects that you have access to. So everybody who is able to log into seqr will have access to a seqr demo project, but you should all also see a Gregor training project. It may have a different letter at the end. If you do not see a Gregor training project when you log in, again, please email through the contact us link at the bottom of the page, and we'll see if we can help get you access during the workshop. There's a high-level overview of each project here, the number of families and individuals and samples in each project. And if you click on the name of the project, that will take you into the project page. At the top of the project page, you'll see a similar high-level overview of the number of families and individuals in the project, as well as the family structures. Now, the genome build it's aligned to, in this case, is build 38. And as you scroll down, you'll see all of the different families within this project. So as a demonstration here, we'll be working on GT9. You can click on the little down arrow next to the family and get some more detailed information with a pedigree, showing you how the different individuals are related to each other. As you scroll down, you can see detailed information on each individual in the family. In this case, we've only really added phenotype details for the proband, GT9_3. There are some structured phenotype terms, as well as some additional info above. So this is a 14-year-old female. She has optic nerve hypoplasia, short stature, congenital onset hypotonia, intellectual disability, juvenile onset seizures, and delayed speech and language development. Some additional notes here: she sat at 1 year, walked at 3 years. So if we're interested in starting to do filtering on this patient, we would go over at the top here and click on the variant search link. And so this page is really where you start your choose your own adventure. So seqr is built to be highly customizable in terms of searches. The great feature of the tool is that we do have some of our kind of standard approaches to filtration built in. So in this saved search header here, if you click on the menu, this is the kind of the order in which we usually go through our typical searches: de novo dominant restrictive and recessive restrictive, and then more permissive versions of both of those searches. So let's start with the de novo dominant restrictive and walk through each of the subcategories. So what this filter does is you can open up each of these headers of different filtering categories. If you click down on inheritance, you can see that the de novo dominant restrictive is asking for 2 reference alleles in all of the unaffected family members and 1 or 2 alternate alleles in all of the affected family members. For pathogenicity, this is where you would search for your previously reported variants. So we would want to return everything that's pathogenic and likely pathogenic in ClinVar. You can see here that I have access also to HGMD. This likely will not appear for you. It's only available for Broad internal users at this time. But as I mentioned earlier in my talk, HGMD is another variant database. Their classification scheme is frankly not very useful, but they do a fairly good job of cataloging variants reported in the literature, so it can be another source of finding previously reported variants. Moving on to annotations, this is where we define what variant impacts we want to look at. For this restrictive search, we typically ask for nonsense, essential splice frame shifts, so those are your loss of function variants, all types of missense variants, and in-frame insertion and deletions. You'll see there are also filter options for structural variants here. Only one of the cases in this project actually has structural data, so this won't be useful for most of the cases, but we did want to point out that if you have structural variant calls in your data, it is possible for seqr to handle those. And again, this is a fairly restrictive search. It's kind of our first pass high-level search, so we don't include a lot of the non-coding or less impactful changes like synonymous variants. There is also the option to filter on in silico predictors. We don't typically use this, but it's nice that it's a feature for anyone interested. I also should have pointed out in this annotation section, one of the in silico predictors, Splice AI, we have worked into this section. We find that Splice AI is a predictor for splice impacts, and we find that it functions pretty well, usually best near canonical splice sites, but we have set a cutoff of 0.2, so this means that any variant will come back if it has a Splice AI cutoff of 0.2 or above, regardless of its impact. So it could be an intronic change. This filter overrides the other annotation filters. For frequency, this is where we'll filter based on our population frequency data. So you can see that we rely primarily on Nomad, though there are other data sets available, such as TopMed. We have here a 0.1% frequency cutoff. Again, this is a fairly restrictive search, and we're looking for de novo and dominant changes, so we expect them to be fairly rare in the general population. Importantly, you're also able to filter on this call set. So this is frequency data from all the samples that were joint called in this project, and is a very useful resource for getting rid of common false positives within your sequencing setup or within the population you're sequencing. And you can see we're a little more permissive with that since we are working with a rare disease cohort. We have certain phenotypes that are enriched, so we allow up to a 1% cutoff for that. Moving on to location. This is where you would have the ability to filter on a specific gene list if desired, not built into the de novo dominant search, but it can be useful to add genes here, especially if you have a proband only case where you'll get a lot more variants returned by this filtering setup. You can either type in gene symbols here. Of note, they have to be in all caps. So for example, if I was going to add CFTR here, there is also the option to use pre-built gene lists, which I don't have any added to this project at the moment, but I can show you later how to add those. You would be able to just select them from the drop down here and it would automatically populate your genes, intervals, regions. You can also search on specific locations and variants if desired. Finally, in the call quality section, this is where we're going to search on our quality metrics. You can see here we have a cutoff of
oops, I just changed that accidentally, 40 for the genotype quality, 20 for allele balance. We also have hard filters from BQSR here. So we have past variants only selected for this particular search, again, which is a restrictive search. These are things that we relax when we go to a permissive search. And again, there are SV quality score cutoffs as well if you have SV data. So once you have all these search parameters selected, you would just click search. But before we go through what the search results look like, I just want to show you quickly what some of the other saved searches do. So if we were, for example, to move to the recessive restrictive, you can then go through each category and see what changes it makes here. The recessive doesn't actually display in this field, but it's looking for compound heterozygous or homozygous changes in the affected individuals that are not compound het or homozygous in the parents. When you do have both parental samples here, it will also determine phasing, so to make sure that when you have two hits in the same gene, they're on opposite copies, one inherited from mom and one inherited from dad. No changes to the pathogenicity section or the annotation section. However, you will notice now that there is the option to do a second hit annotation, so you can define for both variants in the pair what you would like the annotations to look like. This is useful in that you can be a little more restrictive in your second hit, so you don't want to see two intronic variants in trans with each other, for example. You can kind of play with that a little bit there. The frequency for recessive disease is a little more permissive, so we're up to 1% here and 3% in our call set. And the quality metrics are the same. And then finally, if you were to go into the more permissive searches, this is where we're a little more relaxed. For example, in some of the annotations we look at, including some non-coding regions and synonymous variants. And in our call quality, you see we've switched that now to showing all variants, which can be important because there are some real calls that are filtered out when you select past variants only. And it is, of course, fine to customize any of these search parameters to your own needs. So let's go back and start to look at what the search results look like. We'll go back to our de novo dominant restrictive search and just click search again to make sure nothing changed since we did that last. And what will happen once you click search is that you'll see a display below with the first list of how many variants are returned by your search. So in this case, only 6. That's a reasonable number to review. And we can start looking at the variants themselves. So another one of my favorite features of seqr is that it really pulls a lot of information together on each variant to assist in this kind of high-level review process. So you can see each variant is separated by a gray line here. You have on the left information about the gene the variant impacts. You'll see your cDNA and protein change in the middle here, as well as the genomic position in both build 38 and build 37. You can see genotype info at the bottom here. So we see here that our proband is heterozygous. If you hover over, you get some more details on the quality metric there. And that both parents are wild type at that position. It can also be hard to, sometimes if you're analyzing multiple cases, to remember exactly what phenotype you're looking at. So if you hover over the symbol here, you can actually get those structured phenotype terms to remind yourself what you're looking for here. In this column, we have all of our in silico predictors. You can click on show more to see even more of them, and they are generally color coded so that green is benign, yellow is intermediate, and red is leaning towards pathogenic. If you hover over each of these, you will get the cutoffs for the color ranges that we use. So, this particular variant is a missense variant. Most of the predictors are saying it's benign, though it has a slightly elevated primate AI score. This is a database of a predictor, of variation in primates, and so it's suggesting that maybe this position is more conserved in primates and that this may have an impact. Generally, any one in silico tool is not enough to raise my suspicion for pathogenicity. We tend to look for trends in these tools. At the far right, we have our population frequency data. So you can see in our call set, there's only one allele, so that would be this individual. In exec, it's absent, but in nomad, if we hover over, we can see there are 9 alleles in nomad exomes. So that's a fairly rare variant. And if you were to click on this frequency, it would take you directly to the nomad page. That's another great feature of seqr. There are a lot of links that link out directly to information you want to look at. So if I were to click on the genomic coordinate there, it would take me to the UCSC genome browser where there are more tools available, and you can screen through things like conservation of the region. Clicking back here to seqr again, there are links to search for this variant in Google and PubMed to see if there are any existing publications mentioning this particular variant, and it builds a custom search string that has many different ways to label the variant, which is handy. Also at the gene level, there are a number of different links you can use. So we can link out to Decipher, which is a nice resource for protein level information. So for example, it will display protein domains that are known. It's a bad example because there aren't any apparently known for this gene, but also has regional missense constraint predictions from nomads. So you can look back and say, we were looking at position 2747 and look in this region, and you see there's quite a few different missense variants in this region in nomad, which is leaning towards the side that, you know, this variant is less likely to have an impact. Returning again to seqr, there's actually a lot of other resources at the gene level you can look at in addition to Decipher. So if you are to click on the gene symbol, these are a lot of the commonly used resources to investigate gene-disease relationships. So it will display here if there is an OMIM disease association. You can also link directly to the OMIM page at the top. A PubMed search for any publications on the gene, as well as many other resources up here. For the gene level, we, particularly for novel genes that we're investigating, might take a look at the scope of variation within nomad, specifically looking at loss of function constraint, which is also pulled into this page here. And we also frequently look at MGI and IMPC if this is a completely unknown gene, to see if there are any mouse models that could give us some hints to the phenotype. Another very useful feature here is we pull in the gene level expression data from GTEx. So you can see this MUC4 gene is expressed in the colon, esophagus, and mucosa in general, which is pretty typical of a mucin gene. So overall, my impression of this first variant is that I think it's probably fairly unlikely to be related to our patient's phenotype. It's not a gene that seems particularly interesting to that phenotype, and it's a missense variant that, despite appearing de novo in this patient, has fairly mild predictors. Moving on to the next variant, we have this change in GNAI1. So this one is, again, it looks to be a de novo change, absent from both parents. And if we click on to expand our in silico predictors, this one is now looking a little more interesting. So most of them are red, aside from Splice AI, which is wouldn't expect it to be damaging, given that this is just a missense change. So this is intriguing. Looking at our population frequency data, we can see it's completely absent from nomad. This is the only call within our call set, so this is a predicted damaging, extremely rare variant. This is something I would start to look into the gene-disease association for our patients. So again, we're looking for an intellectual disability, hypotonia, seizure, type phenotype with maybe some growth restriction. We can see that this gene is haploinsufficient. And if we hover over this tag here, this is a tag from GenCC, which, as I mentioned earlier, is a database of gene-disease associations. And we can see that ClinGen has actually categorize this variant as definitive, or this gene, sorry, as definitive for a complex neurodevelopmental disorder. So that is very intriguing for our patient, and I would actually click through to this to get some more information from GenCC and from ClinGen here. So this brings you to the GenCC entry for the gene, or again, you get the same information. It's classified as definitive by ClinGen, and you can actually click straight through to the public report from ClinGen summarizing the evidence available for this gene. And something that immediately stands out to me from this summary is that the core clinical features include global developmental delay, intellectual disability, hypotonia, and epilepsy. So that's a pretty interesting overlap for our patient. And this is something I would want to come back to. So another very nice feature of seqr is the ability to tag variants for review later in your analysis process. So if I return to this variant and click this review toggle here, you'll see a tag now shows up that marks this variant for review. If we were to say leave this analysis right now, go back to the family page and refresh it, we'll see that there is now a little colored bar up at this top right corner, which is where all of your saved variants are. If I click on that bar, it now takes us directly back to our list of variants that we've flagged for review later. So this both makes it easy to kind of stop and come back to analysis whenever you need to, but also to collaborate with others on analysis. So anyone else who has access to this project can see the variants that you've flagged for review. There are a number of other tags you can also add to variants if you click on this Edit Tags field. Many of these are specifically designed for our uses in the Center for Mendelian Genomics, where we flag gene level evidence as Tier 1 or Tier 2, but you can see that there's flags for sending for Singer validation, some classification flags as well. We do ask for the purposes of this workshop that you just stick to the review tags, given that multiple different people are working on the same project. So I'll return again to our search for this family. I'm going to go back to the family page and click variant search again. And let's take a look at briefly what a more permissive search might look like. So in this case, let's take a look at the recessive permissive search. So you can see now that we have 102 variants, so quite a bit more. There's a couple things I want to show you in this result. The first is that generally in recessive searches, seqr has a nice way of pairing the compound hat pairs. You can see there's a little indentation here. This is the gene that's impacted, and the pair here where you can see one variant is inherited from the mother and one from the father. A feature that's helpful when you are doing these more permissive searches that return a larger number of variants, or if you're doing a proband only search that is by nature going to return a lot more variants, is that you can actually sort the variants to prioritize which ones you want to look at first. The default is to sort just by genomic coordinate, but you can instead choose to sort by other things like in silico predictions, frequency, or for example pathogenicity. So this would bring anything that's pathogenic or likely pathogenic in ClinVar up to the top of your search queue. Or in this case, if there actually aren't any variants that are classified as path or likely path, it will bring anything with a ClinVar classification up to the top of the queue. I do want to highlight what it looks like when you have a variant that's in ClinVar. You'll see this ClinVar flag at the top here. Also, another flag that we haven't yet seen is this OMIM flag, which shows the gene disease association. It does not mean this particular variant has been associated with a given disease. This is a tag at the gene level. But when either of these are present, you can click directly on the flag to get more information about the gene disease association in OMIM, or to click directly through to the ClinVar entry. That one, as you saw, was a conflicting evidence flag, so you can see there's classifications as uncertain significance by one lab and benign and likely benign by many others in this case. So I'm going to return to seqr again and actually run a slightly different search so that I can show you what it looks like when a variant is pathogenic or likely pathogenic in ClinVar. So I'm going to instead run our de novo dominant permissive search, and I'm going to make a change to this inheritance pattern here. Instead of requiring de novo dominant, I'm going to mark any affected. What this does is require that a variant has at least one alternate allele in the proband, but for unaffected individuals, it doesn't specify any allele state. So this is actually a good technique to keep in mind for situations where you're not sure if one of the parents is affected or one of the other family members is affected, or if there's a concern for incomplete penetrance. So what you'll see when we do this is that our variant count will increase. So we had 6 in the de novo dominant restrictive. We would have had more than that, think about 20 in a de novo dominant permissive in this case, but we're up to 694 with an any affected. And that will kind of reflect what you'll see also if you're trying to do a de novo dominant permissive search in a proband only case. So this is where sorting is really important. And I just wanted to show you that this is what it looks like when you have a variant that has been classified as pathogenic in ClinVar. So it's highlighted red. Alternatively, if it's a benign variant, it's going to be highlighted green. You also get to see the star status in ClinVar, which reflects whether there are multiple labs with similar classifications and no conflicts, and also whether they provide their classification criteria. So when thinking about this particular variant, I'd want to evaluate the gene-disease relationship. So looking at both OMIM and GenCC here, it seems the gene is primarily associated with a hyperlipidemia type phenotype. So that's not something I think I'm particularly interested in with respect to the proband we're looking at, who has more of a neurodevelopmental phenotype. However, if you evaluate the pathogenicity of this variant and consider it pathogenic, you might decide to return this variant depending on your incidental findings policy. So incidental findings are disease-causing variants that are found in an exome or genome that are unrelated to the indication for testing. And these are somewhat controversial in terms of return. There's guidance available from ACMG and other professional societies that can help guide you on this, but it really will be up to kind of how you handle incidental findings, whether you would consider even reporting this variant at all. From my perspective in the analysis of the neurodevelopmental phenotype, this is not a relevant variant for this patient. So were I actually analyzing this case, I would be primarily focusing on these de novo dominant and restrictive searches. And given that we found something already of potential interest, this GNAI1 variant, in our restrictive searches, I would dedicate a lot of time to looking into this variant and gene to confirm that this seems to be relevant to the patient phenotype. I would look into the region of the gene that this variant falls in to confirm that there isn't a lot of, for example, benign missense variant in NOMAD in that position. And then I would probably nominate this one to go on to more in-depth variant and gene curation. Before spending a lot of effort curating the gene and variant, you may also want to make sure first that you believe this is a real variant and that it is a true de novo variant, so not actually present in any reads in either of the parents. So seqr has a nice feature where it can actually integrate visualization of the NGS data directly in the browser. So you could just click on this Show Reads link right next to the genotype calls, and it will automatically open IGV to the position of the variant. Loading can take a little longer. But what you can see is that we'll have data loaded for both parents at the top and then the proband down at the bottom here. And now that that's loaded, we can see that there is an A to G change at this position. It looks like pretty clean reads, about 50-50 allele balance, so this looks like a real high quality call to me. And also reviewing the parental data, I'm not seeing any evidence of the variant in either parent, so that's encouraging. You can open up menu items and customize this view in IGV by looking at read pairs, sorting the reads, and differential ways for coloring the reads. So this is a really nice feature to have built straight into seqr. As you did your digging, you may want to kind of keep track of what you found about the variant. And I do want to point out that seqr does enable the addition of some free text notes to your variant. So I can, for example, enter here that the gene is associated with neurodevelopmental disorder relevant to patient phenotype. I might also want to make a note, for example, that the variant falls in a highly constrained region. And submit that. And the nice thing is that these notes and tags will stick with the variant across different filtrations. So if it comes up again in another filtration approach, you'll know that you've already looked at this change. And you do have the option to edit and delete any notes and tags as needed. One additional feature that's actually enabled in this training project is that seqr facilitates submissions to Matchmaker Exchange for any genes where you are interested in a potential variant, for example, a rare de novo change that looks damaging, but the gene has no existing disease association. You might want to submit that to Matchmaker Exchange, and seqr actually greatly facilitates that. There's actually a button you can select on the family page where you just kind of check any variants that have been tagged for review, and they will be submitted along with any structured phenotype data provided for the patient, and it even enables kind of automatically generated emails to any potential hits that come out of Matchmaker. So just so you are aware, that is a nice feature of seqr. Not enabled in these projects, but there will be a session later on in the workshop about doing Matchmaker Exchange submissions through GeneMatcher. So the last thing I wanted to demonstrate in seqr before we let you loose on analysis is how to add gene lists for use in analysis. So in order to do a gene list or use a pre-built gene list for any case within this project, you first have to add the gene list to the project itself. So I will click this Add Gene List button on the project page, and it will automatically populate with all of the gene list options that are currently public in seqr. You'll see that at the top you also have the option to create a new gene list if desired. But there are already quite a few in here. I'm going to sort them by name here, and we'll say maybe we're looking for an intellectual disability gene list. So scrolling down this list, if I can get my computer to cooperate, we can go down and see all of the many phenotypes that are in here. But as we get to intellectual disability here, we can see that there are options already pre-built from Panel App. So as an example, I'll select this one and then hit Submit at the bottom of that page. And now you can see that the intellectual disability gene list is available within this project. So in order to use this, I would go into the filter for the case we're looking at, and now when I go to the location section here, you'll see in the drop down that I have the ability to select the intellectual disability gene list. It will populate all of the genes from that list in this section, and a nice feature that works for some panel app lists is that it will break down the genes based on the evidence level asserted in panel apps. It's just not available for this list. So if I were doing this gene list based analysis, I would want to make sure also to specify a frequency cutoff. We'll say 0.1 here, and also make sure that I'm looking for variants specifically that are within the proband, so at least one alt allele in my proband. You could also decide to specify that you wanted them to be either absent for the parents or only heterozygous in the parents. And then hit search. And because this is a larger gene list, you're probably going to get a larger scope of search results. So it is a difficult toss-up generally with the number of genes on your list. Generally, gene list searches work the best when you have a more restricted scope of genes to look at. Okay, that's all I think we want to demonstrate in seqr for today.
All right, so in lieu of saving time for questions in this session, I think it makes sense for us to just go straight into the breakout rooms where you will have access to members of the analysis team at the Center for Mendelian Genomics. These are all people that have been working with seqr and doing analysis of exome genome data for a long time, so they're all experts and will be ready to help you out with any general analysis approach questions or seqr questions you have. So we have a suggested order of the cases available in this project, and I will paste this again into the chat so you have access to it as you're working on your analysis. And we'll hand it over to Rolanda to break us into breakout rooms, and I hope you enjoy.